Good morning. Let's discuss on heme biosynthesis. Heme synthesis involves a series of enzymatic reaction collectively known as porphyrin synthesis pathway or the heme biosynthetic pathway. Biosynthesis of heme happens in the mitochondria and cytosol both. It is synthesized by almost all the tissues in the body, especially in the normoblasts, but not in the matured one. Erythroid precursor cells are important site. Nearly 80% of biosynthesis happens in the erythroid precursor cell in the bone marrow and the liver. There are overall 7 steps in synthesis of heme. So one is synthesis of ELA, synthesis of porphobilinogen, synthesis of uroporphyrinogen, synthesis of coproporphyrinogen, synthesis of protoporphyrinogen, synthesis of protoporphyrin, synthesis of heme. Few steps are mitochondrial, few are cytosolic. So glycine and succinyl coenzyme A, they form the alpha linolenic acid. That is the first step. Then it is a mitochondrial step. It goes out, ELA comes out, and ELA converts up into porphobilinogen in the presence of enzyme ELA dehydratase. In the next third step, the porphobilinogen converts up into uroporphyrinogen 3 with the help of enzyme porphobilinogen deaminase. Then uroporphyrinogen 3 converts up into coproporphyrinogen. That is the step 4. Then coproporphyrinogen enters into mitochondria. It converts up into protoporphyrinogen 3. Then protoporphyrinogen converts up into protoporphyrin 3 in the mitochondria. And finally, with the incorporation of iron, this protoporphyrin is converted up into the heme in the presence of enzyme heme synthase or ferrochelatase. So overall 7 steps. 4 steps are the mitochondrial and 3 steps are cytosolic. So step 1, 5. This is the step 1 and 5, 6 and 7 steps. They are mitochondrial and step 2, 3 and 4. They are cytosolic. Here there is more clear account of biosynthetic pathway. So step 1, it involves the succinyl coenzyme A and the glycine. They condense in the presence of enzyme ELA synthase and the coenzyme A is released here, and this enzyme requires the presence of pyridoxal phosphate, and in the presence of this coenzyme it converts up into finally delta aminolevulinic acid. So the enzyme ELA synthase is located in the mitochondria and is the rate limiting enzyme of the pathway, and ELA synthase is PLP dependent, hence anemia is manifested in the deficiency of pyridoxal phosphate. So then step 2 involves the condensation of two ELA molecules 1 and 2 in the presence of enzyme LR dehydratase. Dehydratase means loss of water. So 2 water molecule are lost and it converts up into porphobilinogen, which is a pyrrole. Now the first pyrrole ring is being formed. LR dehydratase is zinc dependent enzyme. So in the next step, step 3, that is also cytosolic step. So then 4 porphobilinogen molecules, these are the porphobilinogen. These 4 molecules, they condense with each other in the presence of enzyme PBG deaminase. Another name for this enzyme is uroporphobilinogen 3. And then 3 co-synthase. Names are very easy to remember based on either on the name of product or either on the name of substrate. So PBG deaminase because there is a loss of ammonia here and it converts up into uroporphyrinogen 3. So a sort of porphyrin ring is now created. The first ring is being created in this step 3. Then and these are the substituent acetyl propionyl, acetyl propionyl, acetyl propionyl and then propionyl acetyl. The sequence is changed it. That's why, because at the third step there is a change in 3 steps are having the altered sequence AP, AP, AP. Then that's how it is named as uroporphyrinogen 3. In the next step, the uroparfinogen decarboxylase enzyme that is going to lose the 4 carbon dioxide molecules, and it converts the molecule uroporphyrinogen 3 into the coproporphyrinogen. So actually what happens, the acetyl group in the substituent site, as a substituent, it loses the 4 carbon dioxide and converts up into the methyl group. So we can see it here in the now acetyl is converted up into the methyl. So it is methyl, propionyl, methyl, propionyl, methyl, propionyl, and propionyl methyl. So this is how it converts up into coproporphyrinogen 3. In the next step, the coproporphyrinogen 3 is converted up into the protoporphyrinogen. And this step requires the molecular presence of NADP. NADP converts up into NADPS. So it is basically the oxidation step. So oxygen is required. So the name of the enzyme is coproporphyrinogen oxidase. Carbon dioxide is lost here. 4 carbon dioxides are lost. So the propionyl group is now converted into the vinyl, which is in the substituent side. These propionyl groups are converted up into the vinyl groups. So the product which is being formed, protoporphyrinogen 3. So we can see it here. This is methyl, vinyl, methyl, propionyl, methyl, propionyl, methyl, vinyl. So this is how the conversion is happening. Finally, all these 2 propionyls are also lost and they are converted into valine. Then this step 2, 3, and 4 step, they are 2, 3, 4 steps are cytosolic. Step 5, 6, and 7, they are mitochondrial. So these step 5 is mitochondrial, step 6 is mitochondrial. So the enzyme is protoporphyrinogen oxidase. It converts the PPG3 into protoporphyrin 3. And the last step where the incorporation of iron will take place, iron is in the ferrous state. So it converts the the enzyme is heme synthase or ferrochelatase, which converts the protoporphyrin 3 into heme. So these are the overall 7 steps in the synthesis of heme. Now move on to the regulation. So regulation of heme synthesis happens at the 2 sites. Regulation in the liver. We will discuss regulation happens in the liver, how it happens, then regulation in the erythroid cells. So ELA synthase, which is the first step enzyme, is main regulatory enzyme as well as the rate limiting enzyme, because this is the slowest step. The heme or hematin control this enzyme by 3 mechanisms. Heme is the final product in the biosynthetic pathway. So if the amount of heme is sufficient, so it will, through feedback regulation, it will inhibit the ELA synthase. Then, so number 1 is the feedback regulation. When there is an excess of free heme, the iron 2 plus 6 state is oxidized to ferric state and converting the heme into hematin, which has lost the capacity to transport the oxygen. Then next is re-separation of ELA synthase. So this is the second way to suppress the synthesis of ELA synthase if the heme is in excess. Third is inhibition of transport of ELA synthase. So because the second step is cytosolic, so transportation of ELA synthase has to happen. So this step is being checked out, and inhibition of transport of ELA synthase from cytosol to mitochondria is also inhibited. Then that is known as compartmentalization, regulation through compartmentalization. Then drugs like phenobarbital, insecticide, carcinogens, they induce the heme synthesis. Barbiturates require the heme containing cytochrome P450 for the metabolism. Then nicotinic acid hydrazide that decrease the availability of pyridoxal phosphate may also affect the heme synthesis. Then ferrochelatase and LRD are inhibited by lead. Then regulation in the erythroid cell, because 80% synthesis happens in the erythroid precursor cells. So it happens through uroporphyrinogen synthase and the chelatase. These are the main regulatory enzyme in the erythroid cell. They regulate the heme formation in the erythroid cell. The cellular uptake of iron also increases the heme synthesis.